Okay, good evening, everyone. So today we are in the week three tutorial session of the NPTEL course Molecular Biology. So, so I will wait for 2 to 3 minutes so that other learners can join the course, um, and then I'll start today's session. Okay, so let's start today's session. Uh, so I hope all of you have gone through the week three video lectures. Namrata, can you let me know if the slides are changing or the slides are visible, right? So earlier I was taking these classes in Google Meet. I have updated the meeting link and I'm taking this session now in Microsoft Teams. So I hope all of you have received the updated link. Mm, okay. Anyway, we will start now. I will start today's session. So yeah, hello everyone. I'm Akash Jyoti Pathak. I'm a PMRFTA for this NPTEL course Molecular Biology. This course is being run by Professor Vishal Trivedi of IIT Guwahati, and yeah, I'm a PhD student in the Department of Biosciences at IIT Bombay, and I'm working in the area of structural biology. Okay, let's start today's session. First, let's summarize what all has been covered in week three, right? So, can someone summarize what all did we learn in week three? In week three, we mainly learned about biomolecules, right? We covered the unit biomolecules. So in biomolecules, we mainly learned about three main biomolecules, that is DNA, RNA, and protein, right? DNA, RNA, and protein. DNA, RNA, and protein. And then under protein, we also learned about the enzymes. Okay, we also learned about enzymes, and in DNA, we we read about DNA sequencing, right? In DNA sequencing, just give me a minute. Okay, we are first summarizing all the topics that we had learned in week three. In DNA, we learned about DNA sequencing and what are the two methods of DNA sequencing. So these are used earlier, right? So now there are more advanced methods of DNA sequencing, next generation DNA sequencing. But the earlier two main methods were used for DNA sequencing. One was this Sanger's method, right? Sanger's method or dideoxy DNA sequencing method. It is also known as dideoxy DNA sequencing method. And another is the Maxam-Gilbert method. Okay, so we learned about these two DNA sequencing techniques. Then we moved on to RNA, right? So, uh, and also we looked about some of the basic concepts of DNA molecules, right? Like the structure of DNA and what are nucleotides, nucleosides, what are the different nitrogenous bases which are present in DNA, what are the chemical bonds present between them. Okay, and then we studied about RNA, right? In RNA, we learned the basic structure of RNA, and then we learned about how we can isolate RNA from cells, right? How we can isolate RNA from cells. So there were two methods by which we can isolate or purify RNA from cells. So one is this ETTP columns we use, right? So because this mRNA, that is, so if you want to purify the mRNA from the cell, so in RNA we have three different types of RNA, mRNA, rRNA, and tRNA. And the mRNA is the one which will be used for translation or which will be translated into protein. Okay. So these mRNA molecules have a poly A tail, right? Have a poly A tail. So if we specifically want to purify the mRNA from the tRNA, rRNA, and tRNA, rRNA. So what we use is we use this DTDP column. So these column, what do they have? They have resins, and these resins are tagged with. So these columns have resins, and these resins. So if you look at this resin, they are tagged with, or they have this DTP conjugated with this nucleotide DTTP. And so what will happen? This adenine, this A tail, poly A tail, will bind to this column, and then these mRNA molecules can subsequently be eluted by increasing the or by changing the buffer conditions, by increasing the buffer conditions. the salt or varying the conditions, we can elute or purify the mRNA molecule. And another method for isolating mRNA from cells is by the trizol method. Okay, trizol method. And then we after so next what we learned about is proteins, right? So in proteins, we first understood the basic building blocks of proteins, which are amino acids. And then we moved further and learned about the different secondary, what is the primary structure of protein, secondary structure of protein, tertiary, quaternary structure of protein, what are the different methods by which you can determine the structure of a protein, right? And like using NMR, cryo-EM, X-ray crystallography. So these methods will help you determine the structure of a protein.
Hello, sir.
Hello, I think my net was off. So till where did, can someone tell me where, till which part were you able to hear?
Polyethyl.
Okay, polyethyl, okay. So all these things you understood, right?
Yes, sir.
Okay. So in today's session, what we will do is we will solve some of the questions, and also I will some of the important concepts which were covered in week three. We will try to cover, and so that you can brush up your concepts, and if you have any doubts, you can clear them as well. Okay. Okay. Okay, so DNA, first biomolecule is DNA. So you all know the structure of DNA. So it is made up of the two polynucleotide strands, right? So it is composed of two polynucleotide chains which are twisted in a double helix. So here in the right side is the structure of a DNA molecule. So this is one polynucleotide strand, this one running from this 3' to 5', and here this is the second polynucleotide strand. And these polynucleotide strands are they are twisted in a double helix, and they're twisted in a double helix pattern and gives the structure of DNA. Okay. So what is the backbone composed of? So the backbone, as we can see here in this figure, that it is composed of this of this sugar and phosphate. So these are the sugar molecules. So in a DNA molecule, the sugar molecule is deoxyribose, and in an RNA molecule, the sugar molecule uh, is ribose. Okay, so if you see the look at the backbone of the DNA, then this backbone is composed of this deoxyribose sugar and the phosphate groups. Okay, so the backbone is composed of alternating sugar and phosphate residues. So this is the sugar and this is the phosphate. So alternately they are arranged. So the sugar, phosphate, and again sugar, phosphate. So they are arranged in an alternate manner. And what are the basic building blocks of DNA? So the basic building blocks of DNA are the nucleotides. So you all know about these concepts. You have already have learned in class 11th and 12th. So the nucleotides are basically have three main components. One is this sugar molecule, the nitrogenous base, and the phosphate group. So the sugar molecule, as you can see over here, right? The sugar molecule, the nitrogenous base, these are the nitrogenous base and the phosphate group. Together these three units or these three things together, they give rise to the is together what we call the nucleotides, right? So so the sugar can be what? It can be 2' 2-deoxyribose as in DNA, or it can be ribose. Okay. So ribose in case of RNA. And what are nucleoside? Nucleoside are, so if you remove the phosphate group, only this nitrogenous base and this sugar molecule is called the nucleoside. It's called the nucleoside. So the bond which is present, what is this bond called? The bond which is present between a nitrogenous base and a sugar is called the glycosidic bond, right? This is called the glycosidic bond. This is called the glycosidic bond, and the bond between the sugar and the phosphate, sugar and the phosphate molecule is called the phosphoester bond. So you, if you look at this structure, then you will see. So this is the phosphoester bond that is formed between this. This is the 3' OH group. So the 3' OH group and the phosphate group give a one molecule of water is released and forms a bond which is known as phosphoester bond. Phosphoester bond. And so if you look at this entire bond, so this is one phosphoester bond and there is another phosphoester bond over here. Okay, so this is one phosphoester bond, this is another phosphoester bond, and together these two bonds are called the phosphodiester bonds. So the phosphodiester bonds are present between the sugar molecule and the phosphate group. Okay. So the glycosidic bond is present between the base and sugar, and the phosphoester bond is present between the sugar and phosphate group. So between the sugar and phosphate group, so one phosphate group is attached to two sugars by two ester bonds. So it is phosphodiester bond. Okay. And if you so you all know they have that these polynucleotide strands, they are anti-parallel, they have polarity. So how this 3' and 5' is named? So if you name the carbon atoms in a sugar molecule, in a deoxyribose sugar molecule, so this is 1, this is 2, this is 3, or 1', 2', 3', 4', and 5'. Okay. So one end of the polynucleotide strand will have the free OH group. So one strand, the strand which is having this free OH group, 3' OH group, is called the 3' end. This is called the 3' end, and the other end of this polynucleotide strand will have a phosphate group which is attached to this 5' carbon, right? So this is the 5' carbon CH2OH. So this 5' this phosphate group, since it is attached to this 5' carbon, it is called the 5' end. So because of this OH, so at one end we can see this 3' OH is free, and at the other end we can see this 5' phosphate group is free. So this gives the polarity to the DNA molecule. So one strand will have 3' to 5' polarity, and the other strand will have 5' to 3' polarity. And so the DNA molecule has an anti-parallel. So the two strands are anti-parallel to each other, right? Okay. Two strands are anti-parallel to each other. So this is the basic of DNA molecules. And then, let's learn about the DNA topology. What do you mean by DNA topology? So if you see so DNA topology means how is the DNA or what is the conformation of the DNA in a cell? Uh, what, what, what type of conformation, or how the DNA remains arranged in the cell. So that is what DNA topology. So there are few terms that comes when we talk about DNA topology. So these are the linking number, then twist and wreath. Twist and wreath. So these are the few terms yeah, which you must understand when we are discussing about DNA topology. So linking number is the number of times one strand is wound around the other. So as we just learned just now that DNA is a double stranded or it has two polynucleotide strands, right? So if this is one polynucleotide strand and this is the other polynucleotide strand, so the number of times this second strand or the second strand. So number of times this second strand twist over the first strand is called the linking number. So if you see over here, so this is one turn. So one, then two, and three. So this second strand has wrapped over the first strand three times. So this linking number will be three. Okay. And another thing is that linking number is always an integer. Okay. It is always an integer. So it will be always in terms of 3, 5, 10 in these numbers. So it is an invariant property and does not depend on the shape of the molecule. Invariant property, why? Because if we see the bacterial DNA, if we see a bacterial DNA, so it is the bacterial DNA has what type of DNA? It is a covalently closed circular DNA, CCC DNA, right? So in an intact covalently closed circular DNA, the linking number will be the linking number will not change. So if the two strands of the DNA are intact, then the linking number will remain constant irrespective of what is the shape of the DNA molecule. Okay. So what is the linking number of a relaxed DNA? How can we calculate the linking number of a relaxed DNA? So the linking number is equal to the number of base pairs by 10.5. So if you remember that the bDNA, so the bDNA has 10.5 base pair per helix. So how many base pairs are present in one helix, DNA helix? So So this is one helix, right? Or pitch as it is called. So this one helix of DNA has how many base pair? It has 10.5 base pairs, right? So the linking number, since linking number is the number of times one strand strand crosses the other strand or wound over the other strand. So usually how will you find it? So it will be equal to the number of base pair, total number of base pairs that are present in the DNA divided by 10.5. Okay, because one turn of helix, one turn of DNA helix has 10.5 base pairs. Okay, so that is why you can calculate the linking number of a relaxed DNA by dividing the number of base pairs by 10.5. Okay, and the linking number can only be changed by breaking the covalent phosphodiester bond. So in the subsequent classes, you will learn about an enzyme called topoisomerase, or you might have learned in week one also about topoisomerase enzyme. So the topoisomerase, there are two types: topoisomerase 1 and topoisomerase 2. So only these enzymes they can cause covalent breaks in the DNA. So only when there is the covalent bonds are broken in the DNA, the linking number can be changed. Okay, and linking number is equal to the sum of the twists and the rits. Okay, so LK equal to TW plus rit. So what is meant by twist and what is meant by rit? So twist is basically the number of times the two strands cross over each other. So basically, if your DNA is relaxed, then the linking number will be equal to the twist. Okay. And Rith is what? Rith is the number of supercoils or undercoils in the DNA in three-dimensional space. So we will understand this in the next slide. When I'll show you a figure, you will understand this better. So twist, it can seem that this twist and linking number are synonymous, but I will explain you how why are these two terms are are sounding similar. So in a covalently closed circular DNA, linking number will be equal to the twist number. And one more thing you have to remember that DNA in most cell in a eukaryotic cell, the DNA is present in a negatively supercoiled form. It is present in a negatively supercoiled form. And how do you say that that the DNA is negatively supercoiled or positively supercoiled? So by this term del LK. So del LK is the difference between the linking number which is present in a coiled DNA or and the linking number which is present in the relaxed DNA. So if you calculate the difference between the linking number in a coiled DNA, so it can be either so in a coiled DNA and the linking number in a relaxed DNA, then if this value comes out to be positive, if del LK comes out to be positive, then we say that the DNA is positively supercoiled, and if this value comes out to be negative, we say it is negatively supercoiled. Okay. So yeah, so this is what I want. This figure is what I was talking about. So if you see, this is a covalently closed circular DNA. So in this figure, you can see that the linking number of so here so in this figure, they have considered an example where the number of base pairs in this DNA is 360. Okay. So now you can calculate the linking number of this DNA molecule because the number of base pairs are given. So if you consider consider this is a B DNA and it has 10.5 base pairs. Okay, so if you calculate this, then roughly what will be the linking number? It will be 36. Okay, so 36 times. So the linking number is 36, which means that 30 that there are 36 times in which the second strand has wound around the first strand. Okay, and the twist will also be equal to, because linking number is equal to twist plus width. So the twist is also equal to 36 because this is a relaxed DNA. So in a relaxed DNA, in a relaxed DNA, the linking number will always be equal to the twist. And okay, now if, for example, if the topoisomerase has act on this DNA and it has cleaved or it has caused a nick in the DNA, and because of which there is a supercoil which is produced in the DNA. So this is the RIT. RIT is what? RIT is the number of how the DNA is coiled in the three-dimensional space. So you see here that how many coils have been introduced. There are 4 coils which have been introduced, right? So that is why 1, 2, 3, 4. So since there are 4 coils, super coils that are introduced in this DNA, so the width is equal to -4, and as a result, the linking number has become how much? So if you linking number is equal to twist plus width, so 36 minus 4, it becomes 32. Okay. So the linking number only changes if there is any breakage in the covalent bond of the DNA. Okay, and that can be done by topoisomerase enzyme, which is present in the cell. Now, for example, this part of the DNA has opened up, and so in this case, what will happen? Now there is no supercoils, or you cannot see any supercoils in this DNA, so the width is 0. So the width is 0, and now and in that case, the linking number. So that is what I was explaining, that the linking number does not change does not change due to the shape or due to the opening of the DNA. The linking number will not change. So here also the linking number was 32, and now when the DNA has opened up, the linking number will still remain 32. Okay. Now try to answer this question. Among the terminologies used to represent the topology of nucleic acids, a dash is always an integer number. So we discussed just now.
D.
Yeah, sorry.
Both
It is A.
twist,
Yeah, sorry, you are saying something.
both twist and
Huh.
width.
Yeah, answer is option A. Okay. So the answer is option A, linking number. So the linking number is always an integer. Okay. Linking number, you remember that linking number is always an integer. And this question, DNA in cells is in dash state. So DNA in cells is just now we discussed that it is in negatively supercoiled state. So since this DNA molecules remain wrapped around by histone octamer complexes, and these complexes result in the DNA um, be, be a DNA attaining a negatively supercoiled state. Okay. So now DNA stability. Let's talk about DNA stability. So how is the why is the DNA structure so stable? So you must have, if you have worked in a lab, you must have heard that DNA remains stable even at room temperature. Or if you have isolated a plasmid, or if you have worked, done any DNA isolation, then you can also keep it at room temperature. It does not the DNA does not get denatured so easily. So what is the reason for the stability of the DNA? So one thing is that the hydrogen bonds, right? So you see that there are in DNA molecules, there are four different types of nitrogenous bases: adenine, guanine, thymine, and cytosine. And in an RNA molecule, there are, in place of thymine, you have uracil. So that is the only difference. In place of thymine, you have uracil. So there are three hydrogen bonds which are formed between adenine and thymine, and there are two hydrogen bonds which are sorry, there are two hydrogen bonds formed between adenine and thymine, and three hydrogen bonds found between guanine and cytosine. Okay. So one reason is the hydrogen bond, but there is one thing you must understand here, that the hydrogen bonds that when a hydrogen bond has to be formed between two nitrogenous bases, the hydrogen. So for example, first you consider that the two polynucleotide strands were separate, right? They have not associated with each other. So when the polynucleotide strands were separate, or when the nitrogenous bases were separate, the nitrogenous bases in the solvent, they were forming hydrogen bonds with the water molecules. Okay, now when these nitrogenous bases has to form on hydrogen bonds with each other, so the hydrogen bonds where between this water molecule and the nitrogenous base has to be broken, and then this and then and they have to form new hydrogen bonds with the other nitrogenous base. So because of this, what happens? Because of this, the entropy increases when a hydrogen bond is formed between the base pairs, as water molecules are displaced from the bases. So the bases in the free form, the bases were very nicely organized, or they were surrounded by water molecules, and because they were forming hydrogen bonds with them. Now, once these nitrogenous bases start forming hydrogen bonds with each other, this hydrogen bond, the entropy, or this entropy increases because the cage-like structure of water molecule, which was formed around the nitrogenous bases, that got broken. Okay. So the one is this entropy also increases, and another, the stability comes from two main effects. One is the electron cloud interaction. So, what is electron cloud interaction? So, the bases interact via transient induced dipole van der Waals forces, which helps hold the helix together. Uh, so, uh, so these you see these double bonds, right? So so there are pi interactions, pi interactions which take place between the double bond, and this is called as the electron cloud interaction. And another is the hydrophobic effect. So what is hydrophobic way, or stacking interaction? So one base pair stacks over the other base pair, and this stacking of nitrogenous bases is possible because of the hydrophobic effect. So by stacking, what happens? The hydrophobic surface of the bases are hidden from water, which is energetically favorable and lowers the overall free energy of the double helix. So these nitrogenous bases are actually they are not very polar structures. So once the base stacking, or the once these bases stack over the other, there are their hydrophobic surfaces get hidden from water, and this leads to the lowers the overall free energy and gives stability to the DNA molecule. Okay. So now you would have to answer this question, phenomena of base stacking observed in nucleic acid is due to. So why is base stacking observed in nucleic acid? So as I was explaining that these nitrogenous bases, they're not polar structures. So what happens once these nitrogenous bases stack over each other? Once the nitrogenous bases are stacked over each other, their hydrophobic nature, because of their hydrophobic nature, the water molecules gets excluded and leads to the stability of the DNA molecule. So yes, the answer is option B. Okay, hydrophobic nature of nitrogenous bases. Okay, now another thing we discussed in the we had learned in the week 3 is the purity of DNA molecules. So can you ensure your DNA is pure or not? So anyway, so you must have isolated a plasmid and then you take its absorbance. So how will you? So for example, you have extracted the DNA from a bacterial cell or from a mammalian cell or any cell you have extracted. Now, since this cell will have lot of other biomolecules, it will have protein, RNA, DNA, and other different types of chemicals might also be present. So how will you ascertain whether your DNA is pure or not? So how is that done? So then in the DNA molecule, the DNA is composed of four different nitrogenous four different nitrogenous bases, right? So these four different nitrogenous bases or these nucleotides absorb very strongly at 260 nanometer. So if you record the UV visible, if you take the absorbance in a UV visible spectrometer, then you will see that these DNA molecules give a peak at 260 nanometer. Okay. So for example, this is your wavelength and you record its absorbance from, for example, 220 nanometer to 320 nanometer. Then you will find that if your sample has the nucleotides or nitrogenous bases, then you will find a peak at 260 nanometer and a short peak at 280 nanometer. Okay. So these points are covered in your lectures. So at 260 nanometer, these nucleotides or nitrogenous bases absorb very strongly. So what we do is, and at 280 nanometer, which biomolecule absorbs at 280 nanometer? Which biomolecule absorbs strongly at 280 nanometer? Protein molecules, right? So the protein molecules absorb very strongly at 280 nanometer and DNA molecules absorb strongly at 260 nanometer. Okay, so if we take the ratio of absorbance at 260 and 280 nanometer, the screen is visible, right? So for assessing the purity of DNA, we take the ratio of absorbance at 260 nanometer and the absorbance at 280 nanometer. So why 280 nanometer? Because the protein molecules absorb very strongly at 280 nanometer, and specifically what type of amino acids? The aromatic amino acids. So though there is a standard or a rule of thumb that is followed. So if the ratio comes between 1.8 to around 2 nanometer or 2, sorry, 2, 1.8 to 2, then we say that the that my DNA is pure. And if it is come, if this ratio comes greater than 2, then there might be RNA contamination. And if it is less than 1.8, then we say that the sample must be contaminated with protein molecule, or my DNA is not pure and it might have protein molecules as well. Okay. So yeah, so the ratio of absorbance at 260 nanometer and okay, so if the ratio is around 1.8, then my sample is pure DNA. If it is around 2.0 or greater than 2, then we say, or if it is, then it is pure RNA. And if it is less than 1.8, then the sample has protein molecules or might be contaminated with protein. So all nucleotides absorb at 260 nanometer. So these are some of the this 8 to 60 by 8 to 80 ratio of the five nucleotides. So you see over here guanine. So the 8 to 60 by 8 to 80 ratio for guanine is 1.15. For adenine it is 4.50. For cytosine, it is 1.51, uracil 4.0, and thymine, it is 1.47. So if you look at the five nucleotides, the uracil has a higher 8 to 60 by 280 ratio. Okay, uracil has a higher 8 to 60 by 280 ratio. It has a ratio of 4. So if we take the absorbance of a DNA molecule and an RNA molecule, then we will find that the RNA molecule will have will RNA molecule will have a the 8 to 60 value for RNA will be more compared to the DNA, because the RNA molecules have uracil nitrogenous base. So since they have uracil, the absorbance or the 8 to 60 value of a single stranded RNA will be more as compared to that of double stranded DNA. So the resultant, so all these nucleotides have different 8 to 60 by 8 to 80 values, right? Then how are we setting this value to 1.8? So basically, this value is set this value is decided based on the. So how is it calculated? It is calculated by uh, by average, by calculating the weight average of all these nucleotide at 260 by 280. Okay, so the resultant 260 by 280 ratio for the nuclear acids being studied will be approximately equal to the weighted average of the 260 by 280 ratios for the four nucleotides present. So the abnormal 260 by 280 ratios, what does it indicate? It indicates contamination by phenol, guanidine, or other reagent. So for example, you must have, if you have gone through the lecture in trizol. So you have studied about how you can isolate the mRNA by trizol method. So by for mRNA amplification or isolation, we use a reagent called trizol. And then there are various other reagents which are used for the purification of RNA, DNA. So if these if these substances like phenol, guanidine, EDTA, trizol, if these are present in our sample, then these because of the presence of these organic solvents, you can the 260 by 280 ratio can be altered. Okay. So now among the following, dash will show the highest absorbance at 260 nanometer. So there are four options here: double-stranded DNA, single-stranded RNA, nucleotides, and protein. So so now you have to answer that among the following of four, which will show the highest absorbance at 260 nanometer. So
Nucleotide.
okay, why nucleotide?
Nucleotides.
So usually
Yeah,
DNA is but highest is that possibility for nucleotide.
yeah, yes, you are correct. So just
That
now we were
that ma-
discussing. Yeah, sorry, what happened to my speakers?
uh, for absorbance maximum.
Hmm. Yeah, so just now we were discussing the 8 to 260 by 880 ratio of these five nucleotides, right? So these are the and the if you for a double-stranded DNA or a single-stranded DNA or single-stranded RNA, that 8 to 60 by 280 ratio will be a weighted average of these four nucleotides, right? So it will obviously be less than the individual nucleotides. So that is why the answer is nucleotides over here. Okay. So you, or if you see over here, so it is uracil for guanine, they are higher than the double-stranded DNA. For double-stranded DNA, we are considering 1.8, or it will be around 1.8 to 2. But for individual nucleotides, it will be more than that. Okay. So the answer is C. Now, ratio of absorbance. So this was there in our assignment, I think. So ratio of absorbance of DNA samples at 260 by 280 nanometer wavelength is 2.1. This observation suggests option is absence of impurities, contamination of protein, contamination of RNA, denaturation of DNA. So just now we learned, right? So what will be the answer? If it is 1.8, then we say that my sample has pure DNA. If it is less than 1.8, then we say that it might be contaminated with protein. Okay. And if it is greater than 2, if it is greater than 2, then we say that my sample is contaminated with RNA, right? With RNA. So answer is, so here the 260 by 280 ratio is 2.1. So this observation, what does it suggest? That it suggests that my DNA sample might be contaminated with RNA. Okay. So answer is option C. Okay, now there were two DNA sequencing methods which were discussed in week 3 lecture. One is this Sanger sequencing method, and another is Maxim Gilbert method. There are few questions related to these in your assignment. So go through these topics well. So I hope these were clear because they were covered in a very broad or covered very nicely in the lecture. So in one is this Sanger sequencing method. So in Sanger sequencing method, you use dideoxy, dideoxy NTPs, right? Dideoxy NTPs. So there are four different reactions. In one of the reactions, you add DDATP. In one of the reaction, DDATP plus other different nucleotide triphosphates. Okay, deoxy, GTP, deoxy, CTP, deoxy, GTP. In another reaction, what will you add? In another reaction, you will add DDAGP or GTP, sorry, DDGTP plus the other nucleotides will be the same. Okay. So now what happens because this nucleotide triphosphate, what do you mean by dideoxynucleotide triphosphate? So you know that the OH group is absent at the 3' position, right? So it is absent at the 3' position. Now if the OH group is also, if you remove the OH group from this 2' position also, then that is called dideoxynucleotide that is called dideoxy sugar. That is called dideoxy sugar. Now, because the OH group is now absent from both 3' and 2', or it is absent from this 3' carbon also, the phosphate or the phosphoester bond cannot be formed. Okay, so for formation of phosphoester bond, you need a 3' OH group and a 5' phosphate group. Right. So if there is a 3' OH group is not present, then the reaction will not be, the polymerase can no longer elongate the strand. Okay. So at that step, the reaction will come to a stop. So in Sanger sequencing, these dideoxynucleotide triphosphates are used, while in Maxam-Gilbert method, what is it? Is a chemical method. So you treat the DNA with different chemicals like DMS, formic acid, hydrazine plus NaCl. So what these chemicals do is they cleave at specific nitrogenous bases. So if you use DMS, it cleaves at G. If you use formic acid, it cleaves at G plus A. Okay. And if you use hydrazine it cleaves at T plus C, and then hydrazine plus salt it cleaves at C. So the fragment. So now you load this in different lanes, and so the band that you see over in this G well in this G lane, and also in G plus A lane, it means that this band is for guanine nucleotide. But the band which is seen only in G plus A lane, that is belonging to the adenine nucleotide. Okay, so that is what is written here. The fragment in G lane is read as G, whereas fragment present in G plus A but absent in G is read as A. Similarly, a fragment in C is read as C, whereas fragments present in T plus C but absent in C is read as T. So now to get the DNA sequence, you will read it from the lowest band to the highest band. In this we will read because the least or the one having the least molecular weight will be present at the bottom, while the one having the highest molecular weight will be present at the top. So you read it from the bottom to the top. Okay. So these chemicals and how and which bonds do they cleave? So these are there in your slides. You can go through the slides, so, and you can solve the assignment questions. Now coming to protein. So the building blocks of proteins are what? They're amino acids, right? So just like the nucleotides are the. Okay, so just like nucleotides are the building blocks of DNA, the building blocks of proteins are amino acids. And so if you look at an amino acid, it has a central carbon. It has a central carbon called the alpha carbon, and this alpha carbon is attached to four different groups. Okay, what are the four different groups to which an alpha carbon or the central carbon? The central carbon is called the alpha carbon, and this alpha carbon is attached to four different groups. And one group is the amino group. The second group is the carboxyl group. Third group is the hydrogen atom, and the fourth group to which this C alpha carbon remains attached is the side chain. Now, if you look at the amino acids, there are 20 different amino acids in our body, in the natural amino acids, and out of and all these 20 different amino acids have these three groups same. So in all these 20 different amino acids, the NH2, hydrogen, and carboxyl group are same in all the 20 different amino acids. The only difference in these 20 amino acids are the side chain. Okay, so the side chain varies. in these 20 different amino acids, and depending on the side chain, what type of side chain is present, these amino acids are classified into uh, these five classes. Okay, so if the amino, if my R chain, if the R side chain is hydrogen, we call it glycine. So all these are non-polar groups, right? Hydrogen, methyl group, then this CH, CH3, CH3. So these are all non-polar groups. So since they are non-polar or aliphatic groups, we these are labeled as all these amino acids: glycine, alanine, proline, valine, methionine, isoleucine, leucine. They come under non-polar amino acids. Now, if my side chain has different has have different ring, have different they have benzene ring or these rings, then we call it aromatic amino acids. If they have nitrogen containing groups, like if they have NH2 groups, then what will it be? It will be basic amino acids. These are basic amino acids because their side chains have NH2 group and they will be positively charged. And if my side chain have carboxyl groups, so if my side chain has carboxyl group like that is seen in aspartate and glutamate, then my amino acid will be an acidic amino acid. And if they do not have charge, but they have polar groups like OH group, or they have CONH2 groups or SH groups. So these are polar groups. Then they these amino acids like serine, threonine, cysteine, asparagine, glutamine, they come under the polar uncharged R groups. So the alpha carbon remains attached. So the building blocks of proteins are the amino acids. There are 20 different amino acids, and in all these 20 different amino acids, the amino acid structure, there is a central carbon called the alpha carbon, which remains attached to four different groups. So, since these four groups to which this P alpha carbon is attached, they are different. They are different in most of the amino acids. We say that this carbon is a chiral carbon. Okay, so you must have learned in chemistry what is a chiral carbon. So chiral what is a chiral atom? A chiral atom is an atom which is joined to. So in case of carbon, we call it a chiral carbon if this carbon atom is attached to four different groups. So in all these amino acids, you can see that this carbon is attached to four different groups. That is methyl group, amine group, and carboxyl group, and then proton or hydrogen group. So these groups are different. So this is the chiral carbon. So since this is a chiral carbon, so it can form different stereo isomers. The stereoisomer is formed are enantiomers. Why is it enantiomer? So since this is a chiral carbon, so it can exist as either L-alanine or T-alanine, and these are non-superimposable mirror images. So they can exist as L form or D form, and since they are non-superimposable, these are called enantiomers. Okay. So there can be either L amino acids or D amino acids, but in humans, what type of amino acids are present on human proteins? Human proteins have L amino acids, right? D amino acids are present in bacterial cell wall. So when we were discussing about peptidoglycan, so the peptidoglycan layer which is present in the bacterial cell wall have D amino acids. Now dash have two chiral atoms. So now can you name also out of the 20 amino acids, can you name two amino acids which have two chiral atoms? So what are the two amino acids? You have to name two amino acids which have, so all these amino acids that you see over here, they have only one chiral carbon, right? This is not chiral. So there is only one chiral carbon. So you have to name an amino acid which have two chiral carbon. So these are the structures of different amino acids. Now, just seeing the structure, can you now find and tell me? Can you search for the amino acid having two chiral carbon and tell me which is the one that is having two chiral carbon?
Tryptophan.
Tryptophan. How are you saying tryptophan? So if you see the structure of tryptophan, so this is the structure of tryptophan, right? So this carbon is chiral, right? Because it is attached to this carboxyl group, this amine group, this hydrogen, and this side chain. Okay, but this carbon, if you see this carbon, this is attached to to two hydrogen atoms, right? It is attached to two hydrogen atoms. So this carbon has two groups same. So this is not a chiral carbon. Okay. So now look at the structure of
Sir,
threonine.
sir,
So if you
sir,
look at the
threonine.
structure of threonine, then you can see that this carbon is attached to what are the four groups? One is this carboxyl group, this amine group, this hydrogen, and this side chain. Okay, and similarly, this carbon is also attached to four different groups: hydrogen, hydroxyl, CH3, and this big group. So these two carbon are chiral carbon. Okay, is it clear? So threonine is an
Yes, sir.
amino acid which has two carbon atoms which are chiral, and other than threonine, there is another amino acid. So out of the 20 amino acids, there are two amino acids which have two chiral carbon. One is threonine and the another is isoleucine. So you look at the structure of isoleucine. In isoleucine also, if you see the two carbon, so this is the first carbon. This first carbon is chiral, right? Because it is attached to this carboxyl amine group, this hydrogen, and this side chain, this big side chain. And this carbon, similarly, if you look at this carbon is also attached to four different groups: CH3, H, CH2, CH3, and this. Okay, so that is why you remember that there are two amino acids out of the 20 amino acids which have two chiral carbon. One is threonine and another is isoleucine. Okay. Okay. So in option you have threonine, so the answer is threonine. But if you do not have threonine and if you have isoleucine in the option, then that is also an answer. Okay. So remember that threonine and isoleucine have two chiral atoms. Okay, so these are the few of the structures of the different amino acids which are given in your options. So if you see alanine, this alanine has only one chiral carbon. Okay, because this carbon has one methyl group, one hydrogen group, one amine group, one carboxyl group. This threonine, if you see this it has two carbon atoms. Both these carbon atoms are chiral. Okay. Because this carbon atom is attached to four different groups. Similarly, this carbon atom is also attached to four different groups. In case of serine, this carbon atom is chiral, but this carbon atom is not chiral because it is attached to two hydrogen groups. So in this case, out of the four groups, the two groups are same. So this is not a chiral carbon. And here also, these are not chiral carbon because it is CH2, CH2. So they have two hydrogen atoms which are same. So they have two hydrogen atoms. That is why these are not chiral carbons. So only threonine and isoleucine have two chiral atoms. So dash is optically inactive. So out of the 20 amino acids, only one is optically inactive. So what do you understand by optically inactive molecule? So what is an optically inactive molecule and what is an optically active molecule? So if my molecule is chiral, if my molecule is chiral, then what happens is, so you have light, right? So the light is vibrating in all in different planes. Now, when you pass it through a plane, this light gets plane polarized and it will now vibrate in only one plane. Okay, so this is called plane polarized light. So when you pass this light through a polarizer, what this polarizer instrument does, it causes all these vibrations to or it cuts away these vibrations and it results in the light molecule vibrating in only one plane. So this is called the plane polarized light. And now when this plane polarized light, if it passes through a molecule, so if it passes through a molecule, so for example, you have a molecule over here, and if this molecule can rotate this plane polarized light in either clockwise direction or in anti-clockwise direction, it's so if this molecule rotates my plane polarized light in clockwise or anti-clockwise direction, then we say that this molecule is optically active. So if a molecule can rotate the plane polarized light in clockwise or anti-clockwise direction, then we say that the molecule is optically active, and molecules which have chiral carbon are optically active. So if so this is basic chemistry you must have learned in organic chemistry also. So if my molecule has a chiral center, then it is optically active. While if my molecule does not have any chiral center, then we say that the molecule is optically inactive. Optically inactive. So optically inactive means molecules which do not have molecules without chiral center. So out of the 20 amino acids, there is only one amino acid which does not have a chiral center. Can you name that amino acid? Do you see the structure of the amino acids? So if you see the structure of glycine, so in glycine, none of the carbon atoms are chiral. So it has only one this C alpha carbon. If you see the C alpha carbon of glycine, then this C alpha carbon, the R group, in case of glycine, the side chain is hydrogen. So since the side chain is hydrogen, so now you have two groups which are same. Okay. So two groups are having hydrogen. The two groups are hydrogen atoms. So in this case, this carbon is not chiral. So glycine is the only amino acid which does not have any chiral carbon, which does not have any chiral carbon. Okay, because in case of glycine, the side chain is hydrogen atom. The answer for this is what? It is glycine. So glycine is optically inactive because it does not have any chiral center. So now, another important concept which was uh, not very well touched upon in the lecture was the charge on amino acid, or what the titration of amino acids, acid-based titration of amino acids. So this is a very important concept, which is important. This is a very important concept of biochemistry actually. The amino acids they are charged molecules. Okay. So so what will be the charge on an amino acid molecule? How is it decided? So if you see, you just now saw the structure of amino acid. It has a carboxyl group and an amino group and an amine group and an amine group. Now in neutral pH, what happens in neutral pH? This carboxyl and this amine group are charged. So this in a neutral pH, this carboxyl and this amine groups remains charged. So when an amino acid lacking an ionizable R group, so the lacking an ionizable R group, what does it mean? That if my R group is or if it is if it is a aliphatic amino acid, aliphatic nonpolar amino acid. So in case of aliphatic nonpolar amino acid, we see that it does not carry any charge. The side chain does not carry any charge. So when an amino acid lacking an ionizable R group is dissolved in water at neutral pH, it exists in the solution as a dipolar ion or Zwitter ion. So Zwitter ion term you must have read in class 12th in chemistry also. So this now when it exists as Zwitter ion, it can act either as an acid or a base. So what is a Zwitter ion? So Zwitter ion means dipolar ion, means it will have both the positive charge and negative charge. Now a Zwitter ion can behave both as acid and a base. Why is it so? Because what will happen if you increase the pH of the solution, so if you keep on adding NOH to the solution, then what will happen? If you keep on adding NaOH to the solution, or if you keep on increasing the pH of the solution, this amine group, this NH3 plus group, it will lose its proton. It will lose its proton, and now it will become NH2. Okay, so since it loses its proton and now it becomes NH2 group, so any molecule which loses its proton is called what? It is called an acid. The acid is a chemical which can donate proton, right? That is one of the definitions of acid, that a molecule which can donate proton is called an acid. So if you keep on increasing the proton, or if you keep on increasing the pH, or if you keep on adding OH into the solution, then this protonated amine group, or this Ne3 plus group, it loses its proton, and now so when this amine group, it loses its proton, it behaves as an acid. Now if you increase the, or if you lower the pH of the solution, if you lower the pH of the solution, or at acidic pH, what will happen? This COO minus group can accept proton. It can accept proton from the solution, and when it accepts proton, now it will start behaving as a base. Okay, so how will it behave as a base? It will accept proton, and what will be the molecular species that will be found? It will be NH3 plus C R H C O O H. Okay, so that is why a Zwitter ion or a dipolar ion can act either as an acid. So it will act as an acid if it loses its proton, and it will act as a base if it accepts proton, and this carboxyl ion gets converted into, or the carboxylate ion gets converted into a carboxyl group. Okay, so that is what is shown here. So Zwitter ion, when it is acting as an acid, it is liberating this proton. This NH3 plus group is liberating proton, and it gets converted into RCH and H2CO minus. And if it accepts proton, then this will behave as a base now, because it is accepting proton, and the what will be produced, what will be generated, RCH NH3 plus COOH. Okay. So now, so this is at neutral pH. So at neutral pH, the net charge is what? So at neutral pH, so if we look in case of this molecule, so if we consider in case of alanine at around neutral pH, because this has an NH3 plus group and a CO minus group, the net charge is zero. Net charge is zero. Now, as you move to lower pH, the this molecule will accept proton, and it will get converted into RCH, COOH, NH3 plus. Now the net charge becomes plus one. Net charge becomes plus one, and as you increase the pH, so as you start increasing the pH, or if you start adding base to the solution, OH groups to the solution, then what will happen? This NH3 plus group will lose its proton, and now the net charge that is present on this amino acid is minus one. The neutral pH, the charge is zero, and at acidic pH, it will be positively charged, and at basic pH, it will be negatively charged. Okay. So why alanine is a diprotic acid when fully protonated? What does this sentence mean? What does this line mean? So alanine is diprotic. So what does diprotic means? Diprotic means that it can liberate two protons. It can liberate two protons. So when it is fully protonated, so in this is the state when this alanine is fully protonated, because here this NH2 group, this nitrogen, because of its lone pair, it has accepted a proton and it has become NH3 plus, and this CO minus has also accepted a proton and become COH minus. So it is fully protonated, and in fully protonated state, now it can liberate two protons. That is why it is diprotic acid when fully protonated. Okay, so because it has two groups, the COH groups and NH3 groups that can yield protons, or NH3 plus groups that can yield proton. Now, let us look at the titration curve of glycine. So what is titration, acid-base titration of amino acid is, so you take an amino acid and you start adding a base into it. So you start adding hydroxyl ions to the amino acid. So what will happen? So At very acidic pH, what very acidic pH, both this NH2 group and this CO minus group will be positively charged. Both the NH2 group and the CO minus group will be, they will accept the proton and they will be fully protonated. So this is the fully protonated state. This is the fully protonated state. And as you continue to add hydroxyl groups to the solution, or you continue to increase the pH of the solution, then first what happens? First, this carboxyl, this proton which is attached to this COH group, first this proton is lost. Okay, so once this proton is lost, then what will be the net charge on the molecule? So this becomes COO minus. When the proton is lost, this will become COO minus, and the net charge will be zero. Net charge will be zero. Here the charge was plus one. Net charge was plus one. And now you keep on adding OH minus or keep on increasing the pH of the solution. Then what will happen? Gradually this hydrogen will also get lost. This hydrogen will also get lost, and you will be left with NH2 now. NH2 CH2 CO minus. The net charge is minus one in this case. Okay. So basically, which proton will be lost first or lost next will depend on the pKa or the pK or the pKa of this group. Okay, so the value of the dissociation constant, negative log of dissociation constant, that will help you determine um, uh, which group will lose its proton first. So if you look at the glycine molecules, so these glycine molecules have two ionizable groups, the carboxyl group and the amino group. The carboxyl group and the amino group, and now this glycine is titrated with a strong base such as NaOH. Okay. Now, when it will be once you start titrating this amino acid with strong base such as NaOH, what will happen? So if you look at this plot has two distinct stages corresponding to deprotonation of two different groups on glycine. So this is the first stage. So if you look at this plot, it has a first stage and then there is another stage. So these two states are because of the two distinct stages are because they are corresponding to deprotonation of two different groups on glycine. So this is NH3 plus group and COOH group. So both are ionizable groups. So what will happen? So at very low pH, the predominant ionic species of glycine is the fully protonated form. So at lower pH means at pH which is less than 2.34. So at pH which is less than 2.34, the predominant species will be protonated, or it will have a plus one charge. It will be the amino acid will be fully protonated. Now in the first titration stage, the COH group of glycine loses its proton. So first this carboxyl group, this carboxyl group loses proton, and at the midpoint of this stage, so this is the midpoint. That is at pH 5.97. So at pH 2.34, the predominant species was protonated, NH3 plus CH2COOH. Now, once you start adding OH, the pH will increase, and at pH 5.97, this carboxyl group will also lose its proton, and it will become COO minus. And now, when it becomes CO minus, this is called the Zwitter ion. Why Zwitter ion? Because the net charge on this molecule is now zero. Net charge is zero. So this is Zwitter ion. And so now if you keep on increasing the pH, or if you keep on adding OH groups, then what will happen? OH minus ions. Then at pH of 12, the predominant form of glycine is this. Now what will happen? This NH3 plus this proton which is present in this amine group, this will also lose, and it will become NH2. And so the net charge will be minus one. Okay. So there are two different things which are two different there are two different species basically, COAH and NH3 plus, and because of these two different species, we are getting two different plots on this titration curve. Okay, we are getting two distinct stages in this plot. At first, this carboxyl always what happens because this pKa or pK of this carboxyl group is lower. First, this carboxyl group loses its proton, and then as you continue to increase the pH, then this amine group or NH3 plus group will also lose its proton and it will become NH2. Okay, and Pi is basically the midpoint. So what is Pi? So Pi is the pH at which the net charge on the molecule is zero. So Pi is the pH. So at pH 5.97, the net charge on the molecule is zero. So this pH is called the isoelectric point. So basically Pi is isoelectric point. So now tell me what will happen at a pH lower than Pi of amino acid. So when my amino acid is present at a pH which is lower than its Pi, then which form will be prevalent in the aqueous solution? Will it be the protonated or deprotonated, neutral, or the sweeter ion form?
Mostly protonated form.
Okay, protonated form. Why? Okay, yes, the answer is correct. It will be in the protonated form. Okay,
Yep.
so because at tell me.
Because of the pH.
So yes, right. So when my pH is very low, at low pH, what at low pH means what? Low pH means that the proton concentration is very high in the solution. So when there are free protons in the solution, and when there are free protons in the solution, and since this amino acid has both this carboxyl group and NH3 plus or NH2 group, it will accept these protons. It will accept these protons and it will get protonated. Okay, so this CO minus, this also gets protonated, and this NH2 because this has lone pair, this also accepts a proton and it gets converted into NH3 plus. So always remember that at pH less than Pi of the solution, at pH less than Pi of the solution, my amino acid will be positively charged. Okay, because all these ionizable groups, they accept protons and hence it becomes positively. Net charge is +1. So it is positively charged. And at pH equal to Pi, what is the charge on molecule? It is 0. And at pH greater than Pi, the charge on an amino acid is negative. Okay. Because as you keep on increasing the OH minus concentration or decreasing the H plus concentration of the solution, then what happens? These ionizable groups, these ionizable groups also lose their protons, and the net charge that remains on the amino acid is -1. H2CO minus. Okay, the answer is protonated. Okay, so now tell me, so if I keep this amino acid at a pH which is equal to the Pi of the solution, or which is equal to the Pi of this amino acid, then in that case, what will be will my amino acid be soluble or will it precipitate? So if I want my amino acid to remain soluble in my buffer, for example, or in a solvent, in any solvent. So if you want this amino acid to be soluble in a solvent or in a buffer, so what should be the pH of the buffer? Should it be equal to the Pi of the buffer, or should it be greater than the Pi of the buffer, or or less than the pi of the amino acid. So you have an amino acid. Now you want this amino acid to remain soluble in a, you want this amino acid to be soluble in a particular buffer. Okay. Now also in that case, what should be the pH of the buffer? Wow, what should be the value of the pH, or what pH buffer you have to take? So should it be equal to the pI of the amino acid? Should the pH of the buffer be greater than the pI of the amino acid? Or should the pH of the buffer be less than the pI of the amino acid? So these are your three conditions. So what type of buffer will you take? For example, the pI is, for
Where pH
example,
is
8. Huh? pH is
high,
high. And if you keep it at low, then what will happen?
so it will
So
be
for example,
protonate.
so if it is protonated, so if the pH is high, then it will be negatively charged, right? And if it is less than the pi, then it will be positively charged. So in both the cases, my protein will be soluble. So if my amino acid is charged, so anything you want to dissolve in water or any buffer or any solvent, it should be charged. It should carry a charge, right? If it does not carry a charge, then how will it form bonds with your solvent, with any ions or any other, it won't be able to form any bonds, so hydrogen bonds or electrostatic interaction. So if there is no charge on the protein, then in that case, my amino acid will not be soluble and it will form a precipitate. Okay. So in both these cases, if the pH is greater than pi, in that case also my amino acid will be soluble. And if my pH is less than pi, in that case also it will be soluble. For example, you want to dissolve your amino acid in water. Okay. So in amine, if your protein is, if your amino acid is positively charged, then it can form hydrogen bonds with the water molecule. Okay. Or there can be this O minus charge which is present on this water molecule. It can also have some electrostatic interaction. But if there is no charge on my amino acid, then my amino acid will form a precipitate and it will not be soluble. Okay. So you can use both. You can use both a higher pH or a lower pH, but you cannot take a pH which is equal to the pi of the buffer, which is equal to the pi of the amino acid. Okay. This is one of the important concepts you need to understand when you are working with proteins. Okay. So always you have to select a pH which is either greater or less than the pi of the buffer. Answer is option A. So the explanation, I've already given the explanation, but let's read what is written over here. So glycine has a net negative charge at any pH above its pi. At any pH below its pi, glycine has a net positive charge. So that is what I was saying. So if pH is greater than pi, it will be negatively charged, and pH is less than pi, it will be positively charged. Okay. So now proteins, let's talk about proteins about protein. So in proteins, we have primary structure, secondary structure, tertiary structure, and quaternary structure. So the primary, what do you mean by the primary structure of a protein? So basically, primary structure of a protein means the amino acid sequence of a protein. So the amino acid sequence of a protein is called the primary structure. Now, what happens now? These amino acids gets gets arranged, or they form a type of conformation, and because of this conformation, they attain a particular structure which leads to different. The structure that is formed is called the secondary structure. So it is either alpha helix, beta sheet, or turns. And now these different secondary structure interact with each other, or they form another conformation, which is called the tertiary structure. So tertiary structure will have a combination of different secondary structures. And now different peptide molecules, when they come together and give rise to a uh, uh, give rise to a complex structure, then we call it quaternary structure. So the primary structure is the amino acid sequence of a protein. Now, what is secondary structure? The arrangement of amino acid residues giving rise to recurring structural patterns. Okay, so when these amino acid arrange in space and they give rise to a structural pattern, they have rise to a structural pattern. So there can be different structural pattern. One is helix, then there can be beta pleated sheets. Okay, like this, or there can be turns or random coils when there is no structure. So this is called the secondary structure. And what is tertiary structure then? So the three-dimensional folding of a polypeptide. So how my protein is folded in three-dimensional space is called the tertiary structure. And quaternary structure is when different polypeptide subunits arrange in space, then we call it quaternary structure. So as you have seen in hemoglobin, if you have read about the structure of hemoglobin protein, then you must have learned about quaternary structure. Okay, alpha to beta two subunits. So there are two alpha and two beta, and these are these different subunits when they come together and form a complex. We call it a quaternary structure. So protein secondary structure. So in protein secondary structure, you have three different secondary structures. One is the alpha helix, one is the beta sheet, and another is the turn. Okay. So these things they were not discussed in much detail in your in the lectures because this is these are a hardcore biochemistry concept, and the scope of this course is mainly deals with molecular biology. But I will, what we will do is just quickly go through this. We will not go into too much detail. So in protein secondary structure, you have three different secondary structure: alpha helix, beta sheet, and turns. So in alpha helix, so what happens? The polypeptide backbone. So if you see a polypeptide backbone, it's tightly wound around an imaginary axis, drawn longitudinally through the middle of the helix, and the R groups of the amino acid residues protrude outward from the helical backbone. So this is the structure of an alpha helix. So in an alpha helix, you see these R groups. So these R groups, they protrude out from the helix. Okay, so these R groups protrude around the helix and they are coiled around. So this is an imaginary axis and they are coiled around an imaginary axis, giving rise to the alpha helix. Now in an alpha helix, what happens? So in one turn of this helix has around 3.6 amino acid residues. So like in DNA, one helix had how many base pairs? 10.5 base pairs. Similarly, in this case, in an alpha helix, one turn. So starting from here to here, there are around 3.6 amino acid residues, and this 3.6 amino acid residues has a length of 5.4 angstrom. Okay, so per residue, you can calculate what is the rise per base pair or per amino acid residue. Okay. So basically 5.4 angstrom, you have 3.6 residues. If you divide it by 3.6, we will get the per amino acid residue, right? Or per amino acid residue, what is the distance between two amino acids, basically. Okay. So what will be? It will be around 1.5, right? Okay. So the distance between two amino acid is 1.5 angstrom. So there are 3.6 amino acid residues, so it is 5.4 angstrom. Now, the most common alpha helix is right-handed alpha helix. There are also a left-handed alpha helix. So these are in great detail in any biochemistry textbook. You will find these concepts, and you must have read in biochemistry. So the structure, how is the structure of alpha helix stabilized? It is by the formation of hydrogen bonds. So what happens in the hydrogen bonds are formed between the NH this NH3 group plus or NH2 group and the OH group. The NH2 group and the OH group forms hydrogen bonds. So the first amino acid or ith amino acid will form hydrogen bond with I plus 4, 4th amino acid residue. So the ith amino acid residue forms a hydrogen bond with I plus 4th amino acid residue, and through the formation of these hydrogen bonds, this entire structure is stabilized. So the structure is stabilized by a hydrogen bond between the hydrogen atom attached to the electronegative nitrogen atom of a peptide linkage and the electronegative carbonyl oxygen atom of the fourth amino acid on the amino terminal side of that peptide bond. Okay, so this NH2 group forms the hydrogen with bond with the with the oxygen atom of the carboxyl group, and and through these hydrogen bonds, this structure is stabilized. And so there are different propensities, or out of the 20 amino acids, there are a propensity that which amino acid can form hydrogen bonds and which amino acid will not be able to form hydrogen bonds. Okay, so a constant on the formation of alpha helix is the presence of proline or glycine residues. You remember that this proline and glycine residues, they have least proclivity to form helices. And why is it so? Now, this why proline cannot form alpha helix is because, or it is not usually present in an alpha helix structure, because this it is not an amino acid, it is an amino acid, and it does not have a free NH2 group. If you see the structure of proline, then you will see that in proline this NH2 group is not free. This is a secondary amine group. And because this NH2 group is not free, it cannot form a hydrogen bond with the oxygen atom of the carbonyl group. Okay. And this glycine is very flexible, and because of this, because it is a very a flexible molecule, and this size is also very small, so it is difficult for glycine and proline to be present on in the structure of alpha helix. Okay, so now answer this question. In alpha helix, hydrogen bond is formed between the dash and dash residue. So just now we discussed that hydrogen bond is formed between the ith residue and the I plus fourth residue. Means if this is the first amino acid, the first amino acid will form amino acid. So there are, for example, there are 4 and 5. So the first amino acid residue will form a hydrogen bond with the fifth amino acid residue. Similarly, the fifth amino acid residue will form a hydrogen bond with the ninth amino acid residue. Okay, so in this way, the hydrogen bonds are formed. Okay. Now, beta sheet. So beta sheet is another secondary structure of protein. So there can be parallel beta sheets and anti-parallel beta sheets. Now, in this in beta sheets, the as you can see, this the backbone of the polypeptide chain is extended into a zigzag rather than the helical structure. Okay, so in alpha helix, the backbone was oriented in a helical organization, while in the beta sheet they are extended in a zigzag fashion. And the hydrogen bonds are so it is not inter. The hydrogen bonds are found between two different polypeptide chain. It is not between a polypeptide chain. It's not within a polypeptide chain. It is between two different polypeptide chain. So it is formed between the adjacent segments of the polypeptide chain within the sheet. The R groups of the adjacent amino acid protrude from the zigzag structure, as you can see. So this is the beta sheet, and you can see the R groups protruding out from the structure of a beta sheet. And the repeat period. So in alpha helix, the repeat period was what? It was 3.6 amino acid residues, right? Or it was 5.4 angstrom. So in around one turn, 3.6 amino acid residues were present, or it was 5.4 angstrom. But in case of the beta sheet, the that the repeat period, it is either 6.5 angstrom or 7 angstrom. So there can be either anti-parallel beta sheet or parallel beta sheet. So in case of parallel beta sheet, the repeat period is 6.5 angstrom, while in anti-parallel beta sheet it is 7 angstrom. And so the repeat period is shorter for the parallel conformation. Parallel conformation. And another thing to understand between the anti-parallel and parallel beta sheet is which one is more stable. Is it the anti-parallel beta sheet or is it the parallel beta sheet? So if you see the pattern of hydrogen bond which is present in anti-parallel beta sheet and parallel beta sheet, as you can see in this figure also that the hydrogen bonds are in line. They are present in line while in the parallel beta sheet they are not in line. So they are in a in a zigzag orientation, so that it does not have a particular pattern. Okay, but in case of anti-parallel beta sheet, they are these hydrogen bonds are in line. So these the anti-parallel beta sheet is more stable compared to the parallel beta sheet. Okay, because of the pattern in which the hydrogen bonds are formed. So these are some points of the beta sheet, and then coming to turns. So turns are present between the, it can be present between two helix. It can be present between, so if there is a helix and then this helix may be connected to a beta sheet. Okay. Um, so between two secondary structures. Okay, so it can be also be present between two helix. So there are different motifs. You might be learning them in higher in even in MSc or the higher classes that these turns they take different patterns, Greek key motif, and there are different types of motifs, alpha, beta, alpha motif. So the basically what are turns? So the turns are the connecting elements that link successive runs of helix or conformation. So the connecting elements that are present between the helix or conformation are called the turns, and the most common turns are the beta turns. The beta turns connect the ends of two adjacent segments of an anti-parallel sheet. So if you see two beta sheets, so these two beta sheets also remain connected by the beta turns. So beta turns are the turns which are present between the adjacent segments of an anti-parallel sheet. And the structure is a 180-degree turn involving four amino acid residue involving four amino acid residue with the carbonyl oxygen of the first residue forming a hydrogen bond with the amino group hydrogen of the fourth. So in turns also it are also stabilized by hydrogen bonds. So the here the hydrogen bond is formed between the first or between the amino group hydrogen of one amino acid with the carbonyl oxygen of the fourth amino acid. Now, now the what is you have to note over here is that in turns the glycine and proline are more favorable because this glycine, first of all, the glycine are very if you look at the burner, the glycine are very small molecules. They are they have a smaller size compared to the other amino acids. And proline, because of its structure, it can easily form bends, or it can be easily present in these 180-degree turns. This glycine and proline residues, they often occurs in tons, while in alpha helix, this glycine and proline have very low proclivity of of being present in alpha helix, while in case of turns, this glycine and proline can often occur in turns, and while glycine can occur in turn because it is small and flexible, and while proline can be present in turn because the peptide bonds involving the imino nitrogen of proline readily assumes the cis conformation. So this cis conformation is helps this amino acids or favors these amino acids to be present in turns. Okay, so dash is commonly found in turns in the protein. So you remember that this glycine, because of the presence, because of its small size, it can easily be present in the. It can be adjusted in these turns, and also another is the proline. Proline and glycine, they are found in turns, while this glycine and proline are usually absent in alpha helix. Now there is okay. Okay, so this is another question. What is the purpose of adding chloroform during RNA extraction using a trizol reagent? So you learned about this trizol, how you can isolate or purify mRNA from the cell by using that trizol method. So what will be the answer to this question? These are the four options. So which option is correct? So this I have directly taken from the lecture slides. So why do you add triazole reagent? Or why do you, what is the purpose of adding chloroform during RNA extraction using a triazole agent? Uh, what is the purpose of adding chloroform? So basically, what happens is once you have lysed the cells and then by your triazole reagent, then the lysate that you will be obtain. The lysate that you will obtain will have all the three different biomolecules. It will have DNA, protein, RNA. Now, when you add chloroform to the reagent or to the lysate, then what happens? There is a phase separation that takes place. So you will get an aqueous phase and an organic phase. Okay, you will get an aqueous phase and an organic phase. So the aqueous phase will have the RNA, and so organic phase will have this protein and other protein and other. We will have mostly the proteins. Okay, and in the middle you have the cell debris. So okay, so the aqueous phase will have RNA. The phase which is present between the aqueous and the organic phase will have the cell debris, and the organic phase will have the protein. Okay, so once you add chloroform, it leads to phase separation, and so the correct answer for this question is to separate RNA from proteins, because once you get add chloroform, this RNA will be present in this aqueous phase, and this protein will move into this organic phase. Okay. So that is what it is even written in the lecture slides also. After adding chloroform and centrifuging, the mixture separates into three phases with the upper clear aqueous phase. So the upper clear aqueous phase contains the RNA, the interface contains the cell debris, and the lower phase is the organic phase, which will have the protein and the lipids. Okay. So now you can isolate the RNA from the, you can easily collect the aqueous phase, and in the aqueous phase, you will now obtain the RNA molecules. Okay. So the purpose of adding chloroform is to separate the RNA from the proteins. Okay. Now let's discuss the last part of today's session. So last part deals with enzyme. Okay. So I will briefly touch upon the enzyme or what is the how does an enzyme catalyze a reaction. Okay, so you know what are enzymes, right? They are biocatalysts. So it helps to basically they are biocatalysts, and they are what? They are proteins. Basically they are proteins, and why are they called biocatalysts? Because they have to, or they increase the rate of rate of any reaction, any metabolic reaction, or any reaction that is taking place inside the living cell. Okay, so first point is all enzymes are proteins, right? Except ribozymes. So ribozymes are the only exception of enzymes, because ribozymes are made up of RNA. So ribozymes are RNA molecules, and they are not proteins. They are RNA, catalytic RNA. Catalytic RNA molecules are called ribozymes. So ribozymes are catalytic RNA molecules. So enzymes may require coenzymes or cofactors. So enzymes are, they are proteins, but other than the proteins, they may have some non-protein part. So the non-protein part of the enzyme, non-protein part of the enzyme is called coenzyme or cofactors. And so there can be different cofactors. Cofactors may be different metal ions like zinc or there can be different metal like copper, zinc. So these are cofactors, and coenzymes can be like NAD, FAD. So these are the different coenzymes. So these coenzymes and cofactors are also may also be required for their catalytic function. Now, many common chemical processes are, why are enzymes required in living cells? So the enzymes, they are required because many chemical processes are unfavorable or unlikely in the cellular environment, such as the transient formation of unstable charged intermediates or the collision of two or more molecules in the precise orientation required for reaction. So for any reaction, what is required? You require that the two molecules should orient itself in a proper conformation. Okay. So if they are not in a precise orientation, then the bonds, the hydrophobic or the non-covalent interactions, these will not take place. So for the formation or for the so there are many different reactions which are unfavorable in the cellular environment. So for these type of reactions, we require enzymes. So what does an enzyme do? It circumvents these problems by providing a specific environment within which a given reaction can occur more rapidly. So basically, what is the function of a catalyst or a function of an enzyme? The function of an enzyme is to increase the rate of reaction. It will increase the rate of reaction. What does it mean? So if a reaction was taking place in 3 hours, when you add catalyst, what happens? In chemistry, you must have read in this reaction will take place in 30 minutes once you add the catalyst. Okay, so similarly, this is the function of enzymes also. So when you add the enzyme, what happens? It increases the rate of reaction, but it does not affect the reaction equilibrium. So the equilibrium is not, if equilibrium is not affected. So these, so if you consider this difference, this delta G, which is the difference in free energy of the substrate and the product, this difference will not be this difference will not be changed when you add an enzyme or in an enzyme catalyzed reaction. Only what will happen, the rate of reaction will be changed or the rate of reaction will be increased when you add an enzyme. Now, so if you see this figure, so in an enzyme catalyzed reaction, so there is a substrate and the product. So so what happens? This substrate is gets converted into the product, and when the substrate is converted into product, a transition state is formed, right? So the what is the transition state? So transition state is a state which is formed for a very small time, and this in this transition in this state the molecule or the species that is formed that has equal probability of moving back to the substrate or forming the product. Okay, so transition state is not a chemical species with any significant stability, and it should not be confused with a reaction intermediate. Okay, so it is not so. For example, enzyme plus substrate is forming ES. So these are reaction intermediate. Okay, or and then it is forming enzyme plus you get the product or any. These are intermediate. So reaction intermediate and transition or intermediates or reaction intermediates are different from transition state. So you cannot say that it is a chemical species or its stability with any significant stability. It is simply a fleeting molecular movement in which events such as bond breakage, bond formation, and charge development have proceeded to a precise point at which decay to either substrate or product is equally likely. So that is what I was explaining. So at this stage, at transition state, the species that is getting formed, it has equal probability of moving in or getting degraded to substrate or forming the product. Okay, and in this stage, what happens? Different bond breaking, bond formation, different charge development or charge will form in that species. Okay. So what happens? How will you define an equilibrium state? So the equilibrium between a substrate and a product is the difference in the free energies of the ground state. So this is the this is the free energy of the ground state of substrate, and for the product, the free energy of this ground state is this point, right? So this is the ground state of the product, and this is the ground state of the substrate, and the difference in free energies between this substrate and the product. This difference in free energy is called this is called the equilibrium. This is called the equilibrium. So the equilibrium between S and P reflects the difference in free energies of the ground state. The difference between the energy levels of the ground state and the transition state is the activation energy. So if you see, so you see this, observe this figure carefully. So if you see in absence of an enzyme, this difference was this much, right? The difference between the ground state of the substrate and product was this much. Now, once you add the enzyme, you see that this difference has not changed. So that is why an enzyme does not change the equilibrium of the reaction. It does not affect the equilibrium state of the reaction. But what will it affect? It will lower the transition state. So what is transition state? Transition state is the difference in the free energy of the substrate and the free energy of the transition state. So activation energy. Activation energy is the difference in the free energy of the substrate and the free energy of the transition state. So this activation energy gets lowered when and enzyme is added. So before the addition of an enzyme, this was the activation energy. Now, once the enzyme is added, the activation energy got decreased. So the rate, how does the so why does the rate of a reaction increase when an enzyme is added? Because this activation energy gets lowered, right? So the rate of the reaction reflects this activation energy. A higher activation energy corresponds to a slower reaction. So the higher the activation energy, the slower will be the reaction. And when you add enzymes, what will happen? The activation energy will be lowered, and hence the rate of the reaction will increase. So the role of the enzyme is to accelerate the interconversion of substrate and product. The enzyme is not used up in the process, and the equilibrium point is unaffected. So the equilibrium point, so this is the equilibrium point of the substrate and the product. This equilibrium point does not get affected when you add an enzyme. Only the rate of the reaction increases. Why does the rate increase? Because the uh, activation energy gets lowered when an enzyme is added. And the enzyme is also, so similarly, enzyme is it is not used up in the process. It will only lower the or it will increase the rate of the reaction. So however, the reaction reaches equilibrium much faster when the appropriate enzyme is present because the rate of the reaction is increased. Okay, so now answer this question. How do ribozymes differ from conventional enzymes? So the this and another question, these are the two last questions of today's session. So tell me
They
the
contain
answer.
RNA.
What?
They contain both RNA and protein.
Ah, they contain both RNA and protein components. No, this is not correct. Ribozymes are made up of only RNA. They are catalytic RNA molecules. They do not have protein. Ribozymes do not contain protein. They do not have any protein component. They are they are catalytic RNA molecules. They are solely made up of RNA molecules. Okay. So that is the answer is option D actually. They can catalyze reactions without protein assistance. Okay. Answer is option D. That is why ribozymes are exceptions of enzymes. They are not enzymes. Are exceptions to enzymes. Why? Because all enzymes are made up of proteins, but ribozymes are made up of RNA molecules. That is why ribozymes are exceptions to enzymes.
So,
Option D. So they can catalyze their own reactions without protein assistance. Answer this question. Enzyme changes dash in the course of reaction.
rate constant.
For which option?
C.
D you are saying.
Rate constant.
Answer is option C. Rate constant. Right. So
Yeah.
So that is what I have been discussing till now, right? So what does an enzyme do? The equilibrium constant is not affected by enzyme. Okay, only the rate of the reaction will increase. And how is the rate of the reaction increases? Because the rate constant increases. So this slide you remember from the video lectures. So you see in absence of the enzyme, the reaction rate was 1.3 into 10 to the power minus 1. Now, once you add enzyme, the rate of the reaction increased to 1 into 10 to the power 6. So only the rate constant of the reaction gets changed when enzyme is added. So you see all these examples of different enzyme, carbonic anhydrase, chorismate mutase, triose phosphate isomerase. So in presence of the enzyme, the rate constant of the reaction will increase. So you see here the rate enhancement, it increased to around 8-fold increase of rate of reaction. This led to around 1 into 10 to the power 9 folds increase in rate of reaction. So the enthalpy change, Gibbs speech, energy, all these will remain same. Only the rate constant of the enzyme catalyzed reaction gets increased when enzyme is added. Okay. Okay, so that is all for today's session. So did you understand all the concepts?
Yes, sir. Sir, I have a doubt.
Okay. Okay, so what we will do in the next class, we will take up more questions, and also if you have doubts, we can take up those questions as well. So thank you for your patient listening. Thank you.
Yes, sir.